Good afternoon, everybody. It is our great pleasure to welcome you to MDF's Meet the DM Drug Developers webinar series. And today we have the great pleasure to welcome Dyne Therapeutics and hear and learn about updates and progress with their clinical trial. A little bit about the Myotonic Dystrophy Foundation. So our vision is that we see a world with treatment and a cure for myotonic dystrophy. Our mission, the mission of the Myotonic Dystrophy Foundation is community, care, and cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. And as many of you know, through our website and through our services, we provide resources and support. So on our website, you find toolkits and publications. You find information about support programs, calendar events. That's probably how you found and access this event. And we host the Digital Academy with hours and hours of recordings and informations about myotonic dystrophy research and clinical care. Next. So today we also want to extend a very special thank you to Dyne Therapeutics as they are the sponsor for our communication and education webinar series at the gold sponsor level. So thank you very much for this and your continuing support. A quick and brief reminder about our annual MDF conference. So the 2025 conference, May 2nd to the 4th in Indianapolis, Indiana. Registration is open for community members and professionals. So please be sure to check out hotel reservation, signing up for sessions and making yourselves familiar with the program. And a pledge, please sign up sooner or later. We have a record number of participants and we are concerned that we are filling up. So please register. Let's go into science and drug development and process thereof. So the very robust drug development pipeline you see here, which again you can find on our website, is really the best measure and indicator of hope. So we have over 20 programs, commercial programs, that have active DM development activities, and as you can see, and 9 are in the clinical testing phase. So this is really what counts for patients and the community, this kind of progress, and that's what we'll hear in a little bit from Dyne. Next one. So again, as a summary and a reminder, clinical trial and clinical development activities, they fall into 4 broad areas, and they follow the essential dogma of biology. So no worries, it won't get any complicated. So from left to right, we have the DNA. The DNA with the repeat is transcribed in a process called transcription to form the RNA. The repeat is also present on the RNA level. The RNA then misfolds and forms these toxic hairpins. And these hairpins, they function really, really well as traps, as molecular traps for important proteins. When these proteins are not available, we have direct molecular defects, and they ultimately result in cellular dysfunction on a whole organ level. So drug activity falls, you see here in area 1. They target inhibition and transcription and gene editing. The big category then focuses on the inactivation of toxic RNA, so interfering and eliminating these hairpin structures. Then we have compensation effects for the trap proteins. So here, trap proteins that are not available are provided through delivery mechanisms. And then lastly, category 4, next. we have a compensation of cellular dysfunction. There, for example, the mixillotine falls in this category. And today we'll hear from Dyne Therapeutics, and they focus in this second category. Next one. So it's our great pleasure to introduce today's panelists. So we have Dr. Ash Dugar. He's the Chief Medical Affairs Officer for Dyne. He brings over 20 years of broad experience across medical affairs, clinical and commercial development, as well as the important component of real-world evidence generation. He's very accomplished. He served as VP and Global Head of Medical Affairs at Sarepta Therapeutics, Intra-Cellular Therapies, Roche, and Pfizer. He began his career at an NIH, where he completed a pharmacoeconomics and outcomes research fellowship, and at the University of Michigan School of Public Health. He also has an MBA from Pennsylvania State University, and his Ph.D. is in pharmacology, also from Penn State. Then we are also welcoming Catherine Beaverson. She is Vice President of Global Patient Advocacy. She guides and implements the engagement with patient advocacy groups like ours to integrate patient expertise and the patient voice in clinical development. She's also very, very accomplished. Catherine held similar roles with Pfizer, Boehringer Ingelheim, Amicus Therapeutics. She is trained as a genetic counselor and has spent over 10 years at New York Hospital, Weill Cornell Medical Center, and Memorial Sloan Kettering Cancer Center. She got her BA from Swarthmore College and her master's degree in human genetics from Sarah Lawrence College. So next. A little bit of housekeeping for the audience. So you can ask questions live. Just put them in the chat, and then we'll sort them. We'll try to answer as many of them at the end. Also, I'm really, really excited because for this webinar, we received a record number of questions. So I'm really looking forward to a robust conversation after the presentations. So without further ado, we want to hand it over to Ash, and welcome to the webinar.
Great, thank you, Andy. That was a really kind introduction. I appreciate that greatly. And before I start, I'd like to thank Tanya, Andy, Cleed, and the entire MDF organization for the opportunity to talk to you today to present our DM1 program. I always say it's a privilege to speak to the myotonic dystrophy community, and we want to thank you for your time and attention today. And as always, with me is as Andy said, our Vice President and Head of Advocacy, Catherine Beaverson. I think you all know her by now. She's been part of the rare neuromuscular community for well over a decade, and she'll be joining me during the Q&A session. So really pleased to have Catherine join us today. Great. Next slide, please. Thank you. I'd like to remind everyone that we'll be making forward-looking statements today, and you can refer to our website for more information. So this is a really important slide. At Dyne, it all starts with our mission: to deliver life-transforming therapies for people living with serious muscle diseases. Our mission is woven deeply into our core values and inspires our employees, our dynamos, each and every day. Next slide, please. So before sharing the most recent data from the ACHIEVE study of Dyne 101 for myotonic dystrophy type 1, we'd like to share a brief video designed to link the multi-system impact of DM1 to the underlying biology of the disease. As Andy walked through just a bit earlier, in DM1, as you all know, there's a higher than normal number of CTG repeats, which leads to something called abnormal splicing or spliceopathy in many genes. And this spliceopathy is thought to drive the multi-system clinical manifestations of DM1. And the video will help us visualize how Dyne 101 is designed to target spliceopathy to address these multiple manifestations seen in individuals living with DM1. If you could start the video, please.
DM1 is a rare disease that impacts you and your family. It affects your body's skeletal muscle, the heart, lungs, brain, gut, eyes, and other organs. DM1 affects people differently, and the extent of impact on these organs is different for each individual. In DM1, mutated genetic material that is at the root cause of disease forms large, sticky loops in the nucleus, trapping important proteins into clumps. These trapped proteins can't correctly cut and paste or splice genetic material required to make necessary proteins. Trapping leads to incorrect splicing, called spliceopathy, which is believed to cause symptoms of DM1, such as fatigue, sleepiness, muscle weakness, myotonia, gastrointestinal symptoms, cardiac arrhythmias, facial weakness, and others. Dyne 101 is a targeted medicine in development as a potential treatment for DM1. It is composed of a fragment of an antibody, or a Fab, connected to an antisense oligomer, or ASO, which is intended to target the root cause of the disease and correct splicing. The transferrin receptor, or TFR1, is found on the surface of tissues affected by DM1. For example, skeletal muscle, heart, lungs, gut, eyes, and the brain. It shuttles iron into cells to ensure their proper function. The Fab in Dyne 101 is designed to attach to TFR1 and uses it to drive delivery of our targeted medicine. Once inside, the ASO component of Dyne 101 moves into the nucleus, where it enables the release of the trapped proteins. Dyne 101 is being studied in an ongoing phase 1/2 clinical trial to determine whether Dyne 101 is able to restore normal splicing in all tissues relevant to DM1, and whether this can lead to improvement in the symptoms experienced by participants in this trial.
Wonderful, thank you. Hopefully that video provides another way in which we can communicate how Dyne 101 is designed and what it's designed to do. Next slide, please. Importantly, our trial has been informed by multiple stakeholders. Prior to starting our Dyne 101 clinical development program, we proactively obtained expert feedback. And that expert feedback came in two forms: from key scientific and medical leaders and from the community living with DM1. We engaged key opinion leaders, or KOLs, from various disciplines, including neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy, among other specialties. Importantly, we gathered this feedback from across the globe. We've also received advice directly from community members living with DM1. They've provided insights based on their living with DM1, as well as experience with past and current clinical trials. This input has been critical in ensuring a patient-centric design and has included, but is not limited to, helping us understand key considerations when families choose a clinical trial, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported and educated throughout the process. And this feedback has been taken directly into the Achieve clinical trial design, and we've also published a paper on the input that we had from the community and how it shaped our clinical trial design. Next slide, please. So let me now touch on the design of the Achieve trial. Achieve was designed with a multiple ascending dose, or MAD, portion to determine safety, the optimal dose, and improvement in a surrogate endpoint called splicing, as you heard about earlier, and functional outcomes. I'm very pleased that we've done exactly that, as you'll see when I discuss the results coming out of the completed MAD portion. ACHIEVE is a 3-part study. There's a 6-month placebo-controlled MAD portion, a 6-month open label extension, and a 96-week long-term extension. Importantly, all patients go on active drug following the 6-month placebo period. We designed ACHIEVE to be adaptive in nature so that we can optimize the dose and dose regimen within the trial, all part of our efforts to move quickly as possible to advance Dyne 101. We've now completed the MAD portion of the ACHIEVE study based on 56 patients dosed, and today I'll discuss those study results as well as the advancement into the registrational expansion cohort to support a planned submission for U.S. accelerated approval. Next slide, please. So this slide provides information on the baseline characteristics of the individuals that have participated in the trial. I won't spend a lot of time on this slide. The key point is that there are generally similar characteristics across the cohorts, which is really important to understand as we look at the data. Next slide, please. So this is a high-level view that we have indeed done what we hope to do with ACHIEVE. The core principle is that with the FORCE platform, we get enough of the ASO, or the antisense oligonucleotide that we designed to attack the genetic basis of disease, to target that core pharmacology of DM1, which is DMPK knockdown in the nucleus and splicing correction, thereby unlocking functional improvements. On the far left of this slide, we show that we effectively delivered the drug to muscle in humans. Next, we see that Dyne 101 indeed knocks down DMPK, and at the go-forward dose of 6.8 milligrams per kilogram, it knocks down DMPK profoundly and more consistently than we saw with the lower dose cohorts. Now, this is critical because it proves that we are engaging the target that we intended to with Dyne 101. And also what you see on the slide is that it corrects splicing as defined by normalization of the CASI, or the Composite Alternative Splicing Index, which measures those key set of genes that are misspliced in DM1 and which we have directly determined through discussions with the FDA can be an appropriate surrogate endpoint for accelerated approval in the US. Finally, Dyne 101 showed broad functional improvement across multiple endpoints, which I will discuss in more detail in the next few slides. Next slide, please. Turning now to safety first. What you are seeing here is our standard reporting of safety that you've seen me present previously. But now this is updated to December 6th of 2024. And the important thing is that there's really nothing new here. We've achieved 855 doses of study drug, representing over 72 years of patient follow-up on study drug. And there are exactly 0 serious drug-related adverse events. Now let's take a look at the broad efficacy measures that we've assessed in ACHIEVE. Next slide, please. So the conclusion from the MAD study is that our go-forward registrational expansion cohort will use a dose of 6.8 milligrams per kilogram. And CASI at 3 months will be used as a surrogate endpoint for predicting meaningful functional benefit at 6 months. So the next few slides drill into the 3-month pharmacology at 6.8 milligrams per kilogram and the 6-month functional results. First, as shown on the left, we showed the CASI correction at 3 months of greater than 20% for the 6.8 milligram dose. And we showed that this is associated with unlocking broad improvement across multiple functional measures at 6 months. In each of these functional measures, the 6.8 mg per kg cohort is always to the right of placebo, as you can see in the teal color, and also to the right of the no effect line shown here at 6 months. So while each of these measures may not be statistically significant individually, we believe the totality of data is indicating a broad and deep impact on function. We've shown these data to our advisors who are world's experts in treating DM1 patients, and they're really excited. They're excited by the breadth of improvement across multiple measures, including measures that look at CNS manifestations like fatigue and sleepiness, and also by the depth of improvement on each of these measures. So we believe CASI correction at 3 months is a relevant and suitable marker for accelerated approval in the US that potentially allows us to get to market quickly with a drug that impacts DM1. Next slide, please. So you've seen this earlier, but I want to emphasize again the relevance of the core pathobiology and pharmacology. We believe that any drug designed to alter that core pharmacology of DM1 should knock down DMPK in order to show functional improvement. And Dyne 101 does this, as you can see on the left-hand part of the slide. And in turn, this is associated with robust splicing correction, which you can see here at 3 months at 25%. So let's see how this is manifesting in our clinical trial endpoints, in the functional endpoints. Next slide, please. So here's myotonia, as defined by VHOT, or video hand opening time of middle finger relaxation. Now, this is a key and early endpoint in DM1, and most experts consider this the earliest clinical marker to potentially respond to treatment. And we show improvement with our 6.8 mg per kg dose, deepening between 3 months and 6 months, with an improvement of 38% or nearly 3 seconds at 6 months. Even with a relatively low baseline compared to the other cohorts, we see a very robust effect on myotonia. Now, while we're really excited about these data, what we've heard from the community is that myotonia is not the most important measure to individuals living with DM1. So let's take a look at what else we've seen in terms of clinical endpoints, starting with strength. Next slide, please. So in terms of strength, what we see is strength gets better at 3 months and deepens further at 6 months. We believe that a 10% relative change from baseline is clinically meaningful in terms of improvement. And one other point, we're not talking about a slowing of decline. We're talking about an improvement from baseline, actually reversing disease progression. If we can move on to the next slide, which shows improvement in 2 functional measures, great. So here, what we see on the left-hand slide is the 10-meter walk-run test. Now, the 10-meter walk-run test is a standard functional assessment in muscle disorders. And Dyne 101 shows an improvement in this measure at both 3 months and 6 months, while the placebo-treated patients show worsening by 6 months. Similarly, the 5 times sit to stand assesses functionality reflecting lower limb strength, trunk strength, but also stamina and balance. And it too shows a benefit, both relative to baseline and relative to placebo at both 3 and 6 months. And this benefit, while with small numbers, is still over a 10% change from baseline, which is clinically meaningful. Let me now turn to one of the highlights of the data for us. If you go to the next slide, please. In ACHIEVE, we also wanted to ask whether Dyne 101 could reach other tissues and potentially impact even more of the clinical aspects of myotonic dystrophy type 1. And this is also a question that we've had from the community. So to assess that, we looked at the MD-HI, which stands for the Myotonic Dystrophy Health Index, a composite patient-reported outcome in DM1. And what we're showing you is that Dyne 101 seems to beneficially impact MD-HI as well. Looking at the MD-HI total score, between the 6.8 mg per kg cohort and placebo, we see a really important 44% change from baseline for treated patients. Now, the MD-High is a composite of 17 subscales, some of which are asking patients about myotonia and strength and function. But we also became very interested in those other subscales, those that might indicate improvement in other aspects of DM1, such as the CNS manifestations. Next slide, please. So indeed, when we look at the subscales of the MD-High related to CNS manifestations, for example, each of them shows improvement on Dyne 101 relative to placebo and relative to baseline. This includes measures of cognitive impairment, sleep, fatigue, communication, emotional issues, and pain. We were incredibly excited to see these data and will continue to evaluate the impact of Dyne 101 on these subscales. We believe that showing improvement on CNS measures such as these will be a very valuable aspect of Dyne 101 in treating patients and treating individuals living with DM1 holistically. Next slide, please. So I'm going to pivot a little bit to looking ahead now. Our regulatory strategy, as I mentioned, is designed to do two things. Move quickly to get this to DM1 patients. We believe we can determine an effect on many aspects of DM1 within a registrational expansion cohort in the ongoing ACHIEVE study. And here's what it looks like. We plan to soon begin enrolling this cohort at 6.8 mg per kg with at least 32, but really up to 48 patients. Splicing correction at 3 months will be the primary endpoint, and we'll have additional functional and patient-reported outcomes measures as secondary endpoints at 6 months. In terms of endpoints, CASI and VHOT are key, and we've powered the cohort to detect statistically significant changes on both. Based on our data, as well as natural history data, we believe CASI is a better predictor of clinical benefit across a variety of measures that matter to patients relative to changes in myotonia. And beyond CASI and VHOT, we've seen clinically meaningful impact on multiple other measures, and we've listed on this slide the other endpoints that we plan to assess. Our belief is that directionally on these additional measures, as associated with splicing correction, can support accelerated approval. Next slide, please. So let me just summarize the update on our DM1 program. We're really excited about the data from ACHIEVE, and these data have allowed us to do each of the things it was designed to do. The data has helped us select what we believe to be the optimal dose going forward, and now we're initiating the registrational expansion cohort. We've demonstrated that splicing correction is associated with broad functional benefit as shown through clinically meaningful endpoints that matter to patients. Based on previous feedback from the FDA, we believe that splicing correction as measured by CASI can be a surrogate endpoint to support accelerated approval. And finally, we've determined that Dyne 101 to date has a favorable safety profile in humans. Next slide, please. So I have to thank all the families, patients, investigators, and their teams for the sacrifices they made to advance therapeutic options, including Dyne 101 over the years. We know there is huge burden that all of you take on to progress the science and advance medicine for every generation. Thank you so much. And again, thank you to Tanya, Andy, and the entire MDF family for having us here today. It was a privilege. And with that, I'll ask Catherine to join and we can take any questions.
Thank you so much. That was great. We always love your presentation. Very, very concise and very, very, very nice. So I apologize to someone in the audience. I made a mistake in answering a question. So CNS stands for Central Nervous System, yes. And that's under central nervous system, we understand that their brain and essentially the spinal cord. So apologies to whoever I clicked off here. So that brings me to the next topic, a little bit of housekeeping again. Please submit your questions. We are trying to get to as many as we can. So what we did is, because we had so many interesting questions submitted beforehand, I propose we categorize them and address them in the first block. We talk about trials, trial inclusion, trial site selection, process timelines, and approval and access to market in very high level terms. So maybe let's start with the first common question. Ash and Catherine, how do you go at a company like Dyne? How do you go about trial site selections?
Sure. Let me take a crack at that. So within this particular therapeutic area, we're really fortunate to have a number of experts around the globe. And so it is very reasonable to start with those who are part of the Myotonic Dystrophy Clinical Research Network, or the DMCRN. This is a group of sites and physicians and their staff around the world that have been working for decades on this disease, both from a basic science point of view and a natural history point of view. They have laid the foundation in terms of how to do these trials, what kinds of outcomes to be looking at in those clinical trials, the understanding of natural history, so that we can contextualize the data that we generate. And so this network, which continues to grow, is the starting place also in terms of looking at clinical trial sites. And they have done an amazing amount of work. And I'll also add that is through the work of the DMCRN, through the work that they have done in NDM1, which is the largest natural history study for DM1. They have helped us get to a place where we know we should be looking at splicing. That splicing is the underlying pathobiology that needs to be corrected to be able to alleviate the various manifestations. It's through their work that the this kind of splicing index endpoint has been derived. And for those of you who love journals and articles and papers and data, I'll just point out a really cool paper that came out a few weeks ago by Professor Provenzano et al. In the Journal of Clinical Investigation. And actually, the title of it is The Splice Index as a Prognostic Biomarker of Strength and Function in Myotonic Dystrophy Type 1. And so this paper is derived from the work of these experts. Catherine, I don't know if you want to add anything.
Yeah, no, great response. I can pretty much sum it up in 3 points, right? You certainly want to choose based on, who has the expertise in the disease. As Ash was explaining, certainly, this is a very complicated condition biologically. So who has expertise in the disease? Who has experience in running clinical trials, right? I think we would all agree that this is a space that is pretty new to working their way through participation in a clinical trial. And then certainly those sites that have a population of folks that can participate in the trial. So I think those are the 3 big ones we might be looking for when we think about site selection.
And Andy, I'm sure you don't want to go down this path too far, but there are also considerations of just, getting through the various country regulations in terms of their ethics committees, IRBs. So this is to get a clinical trial open in a country at an institution, it's a highly defined, highly regulated process. And you need to be able to show that there's the potential for your drug to benefit individuals living with that particular condition.
Thank you. That's actually really important that you made this comment. Thank you. So let's go next step, because we as a foundation, we always have to balance, right, willingness to participate in clinical trials, and then maybe not being eligible, and that being associated with great disappointment. Maybe what would be great for our community to hear some assurance, right, that in a clinical trial, it's really important to have homogeneous patient cohorts. Maybe you can comment on this a little bit, and because that's a question or concern we hear very frequently.
Sure.
So Ash, would it be helpful if I went through kind of what the eligibility criteria was for this study, and then you can kind of take that to a higher level to answer Andy's question as to why, when we define eligibility criteria, we're really looking for a lack of variability across folks who are participating in the trial. And so for folks who are not familiar, and we have spoken to this a couple times, for this trial specifically, it was men or women, right, between 18 and 49 years of age, a diagnosis of DM1, which was defined as a CTG repeat size greater than 100. The onset of myotonic dystrophy type 1 in muscle symptoms at least by 12 years of age, right? Hand opening time, so a functional requirement there, a hand opening time of at least 2 seconds, hand grip strength and ankle dorsiflexion strength. And I think we did exclude the use of assisted devices, right, for the functional tests. And then there were a whole host of other ones, but I think that's pretty much the major eligibility criteria for this study.
Thank you, Catherine. So yeah, maybe to Catherine's point, I'll just maybe take it up a level. So the criteria that Catherine just listed points to a relatively homogeneous group of individuals eligible for the trial. Even with respect to the strength parameters that Catherine just mentioned, we have specified sort of a floor and a ceiling for those strength measures, because we want to be able to, in our phase 1/2 study, really understand what our drug is doing as sort of as specifically as possible in a controlled population. And then what we would imagine what many companies do in phase 3, they go broader. They have a more expansive set of inclusion-exclusion criteria. We, for example, are planning to initiate our phase 3 study this year, and we would have a slightly broader or a broader set of inclusion-exclusion criteria for our phase 3. So it's really to be able to understand what our drug can do, primarily safety first. It's always going to be safety first. And then trying to learn from the phase 1/2 study what measures are most sensitive, what moves in a certain time frame. So we're really learning a lot from phase 1/2. The other really beautiful thing about the regulatory agencies is they want to get great medicines to patients as fast as possible. So there are expedited approval pathways, and in the US, you've heard me reference the accelerated approval path. And so despite a phase 1/2 study, for example, being perhaps a bit more narrow than a phase 3, these kinds of studies have been shown to be conducted and lead to accelerated approvals within neuromuscular. So there is always an opportunity to be able to, yes, be in the phase 1/2, be in the phase 3 if you meet the eligible criteria. But our goal is to be able to get approval as soon as possible, and that's the path that we're on.
Thank you. I really appreciate it that you made it clear. That's for us always a struggle, right? The more homogeneous the test populations are, the less patients we need, the faster it goes, right? And hopefully, the quicker we have a treatment. So
may I just add something, Andy, because I think it links to maybe a question that was asked around this kind of point that we may be excluding certain patients, right? I think Ash said something very important. It's not only important to define what you're going to measure, but will those measurements move in the time frame of your study? So if you pick a measure that doesn't that won't move in somebody who may be well advanced in their myotonic dystrophy, right? You're not going to be able to demonstrate anything around your drug. So yes, it does exclude certain folks, but I think it's, can we measure something within a defined period of time in order to see an effect by a drug?
Great, thank you. So the last two questions on this subject are, and I apologize upfront, I want to tap into your collective crystal balls and the future, because these are also questions we get very, very frequently. So you mentioned phase 3, right? And then there were questions about market. So maybe if you could sketch out in very high level terms, right? Of course, we know this is very depending on the outcome. So what the next steps are, and if everything goes well, how long they will take, and when patients could expect, right, a drug on the market. And please, everyone in the audience, be very patient with Catherine and Ash, because this is a highly speculative answer, and sure answers will only come with data.
So I'm probably not going to give you an entirely satisfactory answer to this. As you would imagine, we always want to be thoughtful about communicating timing of approval and those sorts of information. What I can say is that we are currently working towards initiating our phase 3 this year. Again, we believe that we're designing a study that is not only patient-centric, but we'll have learned from what we have seen in the phase 1/2, to Catherine's great point. We will understand how to prioritize different endpoints that can move in a reasonable period of time that we can show in our phase 3 program. And so as we have more information for the community on phase 3 start initiation, on perhaps moving forward, what some of the other timelines will look like, I'll always come back to the community and talk about it. For now, what I can tell you is we're planning to initiate phase 3 this year. Catherine, do you want to add anything?
Nope, I have nothing to add.
So then maybe I push the last question a little bit, just a little bit further. So let's say you do your phase 3, right, and you show significant differences, right, in the test versus control cohorts. How soon? What are then the usual next steps towards approval? Again, very high level, right? As an education opportunity for our audience.
Yeah. So maybe I'll just sort of rewind a little bit. I want to make sure that this was really clear. So the phase 1/2 study is registrational. It is designed for accelerated approval. And so we will look to file for accelerated approval in the US with the FDA with our phase 1/2 study. So I also don't want to leave the impression that we're going to have to complete a phase 3 before we look to engage with the FDA on an approval path. But to your point, we are planning a phase 3 study that would be, again, placebo-controlled, as one would expect, as a confirmatory study for the accelerated approval path that we're on. And so we don't have all of the details fleshed out. So again, when we have more details in phase 3, I'm happy to come back to this group. But we are pursuing an accelerated approval path for the US with the phase 1/2.
Right. And then, of course, we will take you up on your offer, right, to come back, right, when we have those news, to then sketch out how long it will take to go to market. Excellent. So let's pivot a little bit, and let's talk about the effects of your drug. And let's start first with maybe commenting on clinical trials, future clinical trials for the congenital form, so the early onset form of myotonic dystrophy type 1. Can you again look in your crystal ball and give opinions, right, or perspectives how that would go and how that would happen.
Yeah. So maybe I'll mention one thing which I think was in the questions that came through. We've seen with our platform, our conjugate, that we are able to cross the blood-brain barrier. And so what we have shown in the past is that we're able to get into the not just the superficial layers of the brain like the cortex, but get into the deep brain areas like the caudate and putamen. So the distribution in the brain is very robust with our FORCE platform. On top of that, in our HTFR1 DMS XL mouse model, right, we've shown with our FORCE conjugate that we've been able to actually have an effect on those trap proteins that you described earlier, that we're able to reduce that clumping, reduce that foci in areas like the cerebellum and the cortex of the mouse. And so that is important because that gives us some confidence that we can actually alleviate some of the CNS, central nervous system manifestations of the disease. As I mentioned in the adult study that we're doing now, phase 1/2, we're able to show improvements in fatigue and sleepiness, cognition, and the like. Now, that further gives us confidence as we think about congenital. Now, I want to be clear that what we'd be measuring in a congenital population would be different than an adult population. So, we would have to understand what the appropriate measures are. So in our overall clinical development plan, we absolutely are going to be looking to treat CDM as well. We need to make sure that we are comfortable and regulators are comfortable with Dyne 101 from a safety and efficacy point of view. So we still have to check the box of what we're doing and achieve right now. We have to understand from the regulators that this is, a green light and a go. And then we will think about how we address the CDM population. On top of that, there's fantastic work being done by luminaries such as Nick Johnson and Valeria Sansone in terms of that younger population, the natural history, and what outcomes to be able to assess so if there's a treatment effect or not. So there's also work that we're going to leverage like we did, for the adult population from the NDM1 work that we're going to learn similarly from the congenital and the younger population work that they're doing. So, what I can tell you is we are definitely looking to treat the entire spectrum of myotonic dystrophy. We believe that correcting splicing will serve the entire population suffering from myotonic dystrophy type 1, from the oldest to the youngest. And we just need to learn more from the natural history and understand what outcomes we should be looking at. But we still need to make sure that we've checked the box on the phase 1/2 ACHIEVE study.
Thank you very much. I think it's really important for the audience to hear and to understand, right, that a drug in adults doesn't equal application of the drug in newborns and infants. It's very complicated, and safety always comes first.
Yeah. One would imagine if you look at other neuromuscular programs that have sort of done something similar, gone from either teens and young children to babies, you have to think differently about how you approach it. For example, lower doses, titrations, right? There's a very thoughtful approach that has to be applied. To your point, Andy, safety, you really have to think about how to make sure that there's no safety issues.
Catherine, do you have anything?
No, you guys are
okay. Sorry. I thought, you wanted to
I did too.
No, believe me, I'll jump in when. Thank you.
So let's stick on that subject for maybe just a little while longer. So in your your beautiful video, right, you also showed that other organ systems are affected. You talked about the lungs, right? You talked about the GI system. You talked about the brain. So two-part question. One is kind of sciencey. How do you explain or think about that a drug targeting muscle tissue, right, also finds its way across the blood-brain barrier into the brain, or affects other tissue or organ system like the lung, right, or the GI system? And part two of the question is, how do you think about that with regards to future trials or different trials?
Sure. So one thing that I've talked about in the past and the video covered a little bit is the utilization of the transferrin receptor 1, or the TFR1. And so the TFR1 receptor is ubiquitous. It's highly expressed across skeletal muscle, to your point, including diaphragm, those respiratory muscles. It's highly expressed in cardiac muscle as well. And when we have shown, what we've shown in our preclinical data is that there's actually, even better penetration of our platform to cardiac muscle and diaphragm than other skeletal muscle. It's really a wonderful thing, because it's the cardiopulmonary manifestations that lead to really that very high morbidity and other consequences of DM1. So we really feel good about being able to target those tissues. Similarly, you have the TFR1 highly expressed on smooth muscle as well, on things like the duodenum, jejunum. So it's highly expressed in those smooth gut muscles, and being able to actually alleviate those GI symptoms that we know is huge impact on quality of life of patients is really, really critical. And again, there's an MD high subscale that I didn't show here that shows that we're able to impact the GI symptoms through that patient-reported outcome subscale. So we feel like we can actually get to all of those kinds of muscles because we're using the transferrin receptor 1 targeted fragment antibody. Now, the blood-brain barrier expresses TFR1 as well, and this has been known for a while. So there are other companies that are going after very specific CNS indications, not myotonic, but CNS indications, that are using a TFR1 based approach. And so again, we aren't surprised that we're able to cross the blood-brain barrier given the high expression of TFR1 on the cells. So this all has given us confidence, including the preclinical data, that we're able to alleviate CNS related symptoms. Did that answer your question?
Yes. So I have one more follow-up question with regard to that, and that's also.
I'm sorry, I was troubled.
Yeah, let me just finish that, and then Catherine, you get the next comment. So do you see then opportunities for future trials, right, where you would, for example, only focus on patients with cardiac presentations or CNS, so brain or central nervous system presentations?
Yeah. So it's a great question. I think we have to think about that a little bit more. I'm hopeful that between our phase 1, 2, and phase 3, we'll be able to demonstrate holistic benefit. That said, we are acutely aware of, and we think a lot about how to specifically show efficacy in cardiac muscle. We think that would be, quite an interesting approach. And I don't know if it's a standalone study or if it's within an expanded population in phase 3, for example. So, but your point is really well taken. How can we have more evidence of targeted efficacy in really specific muscle like cardiac muscle. So we're actively talking about it, what endpoints to use in our phase 3, what we would want to look at with those endpoints, through EKGs or other kinds of lab assessments, if you will. So I hope to have more information on that for you, but it's really a big point of discussion. And I mean that from all the aspects you're saying, the cardiac measurements for efficacy, respiratory, like FVC percent predicted, FEV, what should we be looking at? So we're having those discussions. And then to your point, not just through MD high looking at CNS, but specific subscales on fatigue, on excessive daytime sleepiness, even on cognition. So we're having all of these conversations in depth to be able to understand, how we can design our trial to be able to evaluate the broad manifestations of the disease. So you're spot on. It's like you're in our team meetings, Sandy.
Catherine, sorry, the floor is yours.
No. I'm going to add, because it just builds on what the question was, Andy, your great insights there, and what Ash was saying is, there is a balance, right, between how much you measure in a study and how you get to meaningful benefit. And so, we had an awesome conversation recently with the members of the community to kind of put that out there. What is meaningful benefit, right? And kind of throw out on the table different ways to measure that. But then when you try and fit it into a day, are people willing to spend 11 hours to get all of that in? And maybe they're not, right? Physically, they can't, and probably psychologically, they don't want to. So it is, to Ash's point, an active conversation right now to say, how do you take those things that are meaningful to folks, right? Measure them and then package them into a clinical trial that, answers everything. We probably won't get there, but we have to get to what's important. And they were super, super helpful on questions of what should we be measuring. So thanks.
Promise you, we won't have an 11-hour day. Promise we won't do that.
You're right. No, not happening.
So I wanted to end with two more questions, but I'll scrap the one how your drug performs relative other drug development efforts, because that's really not fair, right? Because Dyne knows the Dyne drug and the others know the other drug. So let's scrap that one. But maybe let's end with let me end with a little bit of speculation with regards to potential lessons learned from your study and focusing on DM1, and what can we learn from such studies with regards to DM2, so myotonic dystrophy type 2. And I know that's hard, and again I want to tap into your
crystal ball there.
Crystal ball.
The batteries have run out of the crystal ball, just so we're clear. So I'm going to maybe try to answer that in a couple of ways. And I'm also happy to talk about differentiation, because I know that's a common question. So what have we learned, right? So what we have learned, which, I didn't show today, is that, at least for DM1, that the correlation between splicing correction and the functional assessments is robust. We presented that in other forum earlier in January, but we're able to show that if you improve that the change in splicing is predictive of clinical benefit. And that's really important, because we've heard from the community that myotonia is bothersome, but it's really all the other stuff: sleepiness, fatigue, the weakness, the mobility issues, that's most impactful. So the fact that we are able to show the correlation between this biomarker of splicing correction and functional improvement, it's a really important learning across the board. And it's really important for us to be able to communicate that and show that. I think from our perspective, obviously the target for DM2 is different than the target for DM1. And so what we've learned is the key component is delivery. And that is why using the transferrin receptor is so important, because we're able to deliver a variety of different genetic payloads to attack the basis of the disease using that particular avenue of the FORCE platform. So again, we try to apply these learnings across multiple programs, and we're going to look to do it in other in other areas, and perhaps even DM2. But we've seen it in Duchenne, in FSHD, in Pampa, and so we really feel like we have something here with the TFR1 receptor mediated delivery of different genetic payloads.
I think that's a wonderful summary, right? And bridging the divide, so to speak. So with this, I think we are past the top of the hour. I want to, on behalf of the foundation and of the entire community, thank you so much. As always, thank you for spending time with us. Thank you for answering difficult questions, right? And thank you for your energized crystal ball and your perspectives.
Thank you. Again, it was really a privilege being here. Thank you so much.
So if you, to the audience, if you like today's program and the speakers, which I hope you do, please consider showing your appreciation with a donation. And then we also learned today, right, that clinical trials are important. So if you are living with DM, please participate in our Myotonic Dystrophy Family Registry. Only together can we make change. And with this, thank you to all of you in the audience. Thank you for the team. Thank you, Cleet and Genevieve, for working in the background. And until next time, thank you.